Welcome students to MBBS Online Lecture 2021. The topic for today's lecture is about RH blood group system. As we have discussed in the previous lecture about the different blood group types, and we have discussed in detail about the ABO blood group system. RH blood group system would be easier if we are well aware with the ABO blood group system. It is also has the same physiology as of ABO blood group system. So let's discuss it. What does RH refers to? RH is the most important blood group system after ABO in transfusion medicine. As I have previously discussed it, that the two blood group system are most important, ABO and RH, because they are very responsible for the transfusion reactions. So one of the most complex of all RBC blood group systems with more than 50 different RH antigens. Okay, rhesus factors, rhesus factor, RH factor. People are divided according to the presence or absence of RH antigen agglutinogen on RBC's membrane. As I have discussed in the ABO blood group system, the people whose RBC having A antigen on their surface, they are referred to the blood group A. The same case is with RH factor. The people who have RH factor, RH antigen present on their surface of RBC, they are termed as RH positive. While people not having RBCs or RBCs not having D antigen, yeah, it RH antigen is also called D antigen. Okay, so people not having D antigen are termed as RH negative. RH positive is distributed in 85% of the people, while RH negative is the case in 15% of the people. This is the distribution of RH positive and negative. RH blood group system. Present positive, if RH factor is present, we term it RH positive, or if it is absent, no RH antigen on the surface of RBC, so it is termed as RH negative. Here it is well diagrammatic representation of RH positive and RH negative. See, there are D antigen on the surface of RBC. They are having RHD antigen, that is RHD gene on the loci of chromosome that may result in the transcription and translation in formation of RH antigen, that is D antigen, that is readily exposed on the surface of RBCs. While in RH negative person, there is no RHD gene present on their chromosome. So no D antigen is exposed on the surface. This is too much detail. We are not concerned with this. RH blood group system for classifying blood groups according to the presence or absence of the RH antigen. RH positive and RH negative, I have discussed it previously. Often called RH factor, it is also called RH factor, which RH antigen is also called RH factor on the cell surface membrane of cells that are erythrocytes, red blood cells. Okay. Compatible blood type donors, A positive, donate blood to A positive and AB positive, can receive blood from A positive and negative, O positive and O negative. These are the chart that refers to the blood transfusion, can, to whom blood person can donate blood to and can receive from. Okay. Testing for A positive blood with an ABO plus G card. As we have discussed previously about the laboratory test about the blood typing, that if the clumps are found with a particular antigen or particular antibody, so we are determining identifying the particular blood type by observing the formation of clumps in the sample. Just like this, if it is blood is A positive, we are having a sample of A positive blood, and we are having antigen A, antigen B, and antigen D. So with antigen A, they the clumps are formed within the sample. So we are sure that this blood group is blood group A. While with anti-B, there is no clump formation. Agglutination does not occur in this one, so it is clear liquid. While with anti-D, the clumps are formed. So it means that the RH factor is present. Okay, I forgot to mention the agglutinin present in term of RH factor. RH positive blood, RH positive means it has having RH antigen. So it does not have RH, or we say anti-D antibody. If a person is RH positive, so he or she has not having anti-D antibody. And if person is RH negative, so he or she not having, sorry, he or she having anti-D antibodies in her blood. So if the sample would be A negative, so there when treating with anti-D antibody, no clump would be formed. But here with the treating with anti-D antibody, we are observing clump formation, agglutination. So we are pretty sure about that this blood group is A positive. This is testing for A positive blood or RH positive. Positive blood versus negative blood means RH positive and negative. Positive blood is a type of blood in which the red blood cell contain. Okay, this is the only definition. Can be found in 85%, while it is 15%, determined by D or D and D small d genotypes. I mean, if D, capital D and capital D are present, so 100% chances are there that it is RH positive. In D and D are present, it is also RH positive because G is dominant character, while determined by small d genotype. Antigen D is present. I have discussed it. While in RH negative, antigen D is not present. While in RH positive, anti-D antibodies are absent, while in RH negative, anti-D antibodies are present. So can only be transferred to an individual with positive blood, mean positive blood to positive blood, while can transfer to an individual with positive blood. RH negative can also be transferred to positive person carrying positive blood. Okay. The term RH is derived from the use of blood of rhesus monkey. It's the basic test for determining the presence of the RH antigen in blood, human blood. The RH blood group was discovered by Karl Landsteiner, who also discovered ABO blood typing, and A.S. Wiener. Okay, let's discuss further. The RH antigen carriages are danger for the RH negative person who lacks the antigen. If Rh positive blood is given in transfusion, adverse effect may not occur the first time Rh incompatible blood is given, but the immune system responds to foreign Rh antibody by producing anti-Rh antibodies. Basically, what happens in Rh negative blood, person does not have anti-D antibodies frequently. These are developed over a long period of time. Unlike APO blood group system, where antibodies are formed frequently, in Rh negative person, anti-D antibodies are not as much in concentration or are at zero concentration. When a person is exposed to Rh positive blood, so in that case, the immune response is triggered and it results in the formation of IgM and IgG antibodies that are of Rh and that are termed as NTD antibodies that may result in agglutination or hemolysis when frequently blood transfusion occurs, just like Rh positive to Rh negative. And another case is a similar hazard exists during pregnancy for the Rh positive offspring of Rh incompatible parents. When the mother is Rh negative and the father is Rh positive, the more chances are fetus would be Rh positive because the dominant characteristics are from father. The first child of such parent is usually in no danger unless the mother has acquired anti-Rh antibodies by virtue of incompatible blood transfusion. So I am discussing this case. This is called HDN, hemolytic disease of newborn, or we also called it hemolytic disease of newborn. Okay. In this case, in first pregnancy, the mother is Rh negative, while the baby is Rh positive, as the father is Rh positive. So what happens during the labor process when the baby is about to birth? So some of the blood from the baby, Rh positive blood, is transferred from placenta towards the from umbilical cord to placenta and ultimately to mother circulation, maternal circulation. So since mother is having Rh negative, so she also has NTD antibodies. When NTD antibodies comes in contact with the Rh positive antigen or D antigen that is transferred from umbilical cord to maternal circulation. So there is no such response or there is no such danger in first pregnancy because baby is born already born. But mother has acquired anti-Rh antibodies. Means mother mother has having circulating anti-Rh antibodies. But when the second pregnancy occurs, and the R anti-Rh antibodies, or we say NTD antibodies, transfer from maternal circulation to the baby circulation through placenta, because in this time secondary immune response occur that results in the formation of IgG antibodies that are very small and can cross placenta easily. So these antibodies then attack on fetus blood that has having anti that has having D antigen and may result in hemolysis of the blood cells. Okay. So there is also discussed the RH are highly immunogenic, and most of the RH antibodies should be considered as potential cause of hemolytic transfusion reaction and HDN, hemolytic disease of newborn. D antigen after A and B is the most important RB in transfusion practice that I have discussed later previously. Okay, let's come to the cross matching. What is cross matching? Cross matching is one of the most important serological procedure pertaining to blood group serology and is fundamental procedure responsible for safe blood transfusion. Basically, cross matching is an antigen antibody reaction, a correct interpretation of which is the most essential preliminary step in the practice of safe transfusion of blood. By cross-matching, we are able to detect atypical and clinically significant antibody, mostly IgM and IgG present in recipient serum or in donor serum. Also, by autocontrol, we are able to detect autoantibody in patient himself. This cross-match test is carried out to ensure that there are no antibodies present in patient's serum that will react with donor cells when transfused, unless there is urgent need for blood. A cross-match must be performed for red cell transfusion. So basically, in this type of procedure, red blood antibodies, or we say of a donor blood, plasma of a donor blood is reacted with the red cells of recipient blood. Plasma of donor and the red cells, RBCs, pegged RBCs of recipient blood are cross-matched in vitro, mean outside the body. So if agglutination occurs, so transfusion is suspended there. But if no agglutination occurs, the transfusion is safe to go. So this is a very much important procedure by to prevent hemolytic transfusion reactions also, and also autoantigen and autoantibody. Donors, sorry, donors red cell plus patient serum. Sorry, I have termed it incorrectly. This is the right. Donor red cells and patient serum are reacted together in vitro. Thank you for watching this video. Hope you all like this video. Don't forget to like, share, and subscribe, and share it with your friends. Thank you.